Good day, Junior Techies. Welcome to this video where we're going to be looking at an exam practice question. I've included this question as question 5 of the worksheet. This is taken from a previous exam paper, and I want to work through this with you to show you exactly what could come out as an investigation in in one of your assessments at the end of the year. Now, you will get an investigative question of some sorts in your final exam, okay? And it's normally the question that a lot of students struggle with. So it's super important here for us to go through and practice questions like this. So I'm glad you're here with me. My name is Mr. P. I'm going to take you through this question. You'll see that I'm also responsible for some of the other videos in this section on cellular respiration. But let's dive right into this investigative question. So question 5 of the worksheet, which is our first exam type question, we need to understand the context. So I've only put the context of this question up on the screen right now. Here you can see that it was an investigation that was carried out to find out if living organisms give off heat energy during cellular respiration. So heat energy is what I'm looking for. Cellular respiration, whether it takes place or not, is actually going to be the context that I'm going to be using. So the procedure here in understanding of how this experiment has taken place. Remember, we're not designing this. This is an experiment that has taken place. Scientists have done this, and this is how they did it. They took 20 pea seeds. Pea seeds were divided into 3 groups. 40 seeds were treated, so it's 3 groups of 40 seeds, my apologies, and then each of these groups of 40 seeds were treated in the following way. Flask A, 40 pea seeds were soaked in water at 18 degrees for 24 hours. Then they were placed in a sterilized vacuum flask. Now there's a reason the flask is sterilized. We'll talk about that a little bit later. But you'll see the context for flask A, B, and C is that after soaking, the temperature of 18 degrees Celsius and the duration of 24 hours is the same for each one. So you should already be thinking about what variable that may be. Anyway, let's look at question, or should I say flask B. So here we've got 40 pea seeds soaked in water and then boiled, cooled to 18 degrees Celsius, then placed in the sterilized vacuum flask, just as flask A. Flask C, 40 pea seeds were soaked in water at 18 degrees Celsius for 24 hours, washed with a mild disinfectant. Now, what would a disinfectant do? You've got to ask yourself that. Normally, what does disinfecting a surface mean? Okay, and then they were placed in a sterilized vacuum flask as well. So now we've got some context to what's happening for each of these groups of 40 pea seeds in this investigation. Right. Just to highlight a few points, there we go. Okay. So moving on now to the actual setup of what this would look like. This is just one of the flasks. Remember, we've got 40 pea seeds in 3 different flasks. This is how each flask would have been set up. I've got the vacuum germinating pea seeds, or depending on the results, they could maybe not germinating. Then I've got a thermometer. Thermometer is going to give me an indication of the change in temperature, because that is what I'm measuring. Let's have a look at our results table here. So these results were given to us. The temperature measured inside of the 3 flasks, A, B, and C, and they were observed every 12 hours for a period of 3 days and recorded in the table below. So flask A, B, and C, and the time in hours, because times, or should I say the measurements were taken every 12 hours for 3 days, you can see there that the time in hours has a 12-hour interval between each one. So that's a pretty standard or constant variable for this investigation. The fact that the measurements were taken at the same time for each of the flasks. So at the onset of this investigation, you'll remember, we started each flask at 18 degrees Celsius. Flask A, after 12 hours, measured 26 degrees Celsius. Flask B remained at 18, and Flask C is at 20 degrees Celsius. So already we can make some sort of, or draw some sort of a conclusion simply after the first 12-hour interval. What's happening in Flask A? Is cellular respiration taking place? Well, if heat is being released, there has to be some sort of chemical reaction taking place. So I would say definitely in Flask A. Flask B, well, temperature remained constant at 18 degrees, so nothing really taking place in Flask B. Flask C showed some increase in temperature, but not quite as much as Flask A. Now, how would we account for that? What was the difference between Flask A and Flask C in our context? So if you go back to that question in your worksheet, have a look at the context of Flask A and the context of Flask C, and see if you can identify the difference. Right. Just some things to remember. This is only one of the three setups. You'll see that Flask A was normal, washed in water, placed in a flask 18 degrees, and that shows the temperature change over 72 hours. Flask B was boiled. Or should I say those pea seeds were boiled. So what did the boiling do to the seed? This tells me it actually removed the seed's ability for cellular respiration. So this actually destroyed the seed completely. So in other words, it killed the seed. Flask C, this is where we had the sterilized pea seeds. Sterilized. So sterilization would mean we are removing all of those microorganisms that could have been present on those seeds. Do microorganisms respire as well? Well, they do. Could this add to the temperature change in that flask? Well, potentially. So let's have a look-see at what the questions are going to ask us. First thing, 5.1. Questions always deal with variables, so we can expect the independent, dependent variable question more often than not. So here, it asks us for A, the independent variable, B, the dependent variable. I like to deal with the dependent variable first before I approach the independent variable, because it makes me identify the independent variable so much easier. So dependent here is the heat given off or the change in the temperature. So what does that actually give an indication of? Whether or not cellular respiration has taken place. Okay, so that's the first thing to look at. If we look at the independent variable, what causes this change in heat? Whether or not the cells were respiring or not. In the case of Flask A, we saw an increase in temperature. Did cellular respiration take place? Yes. What about in Flask B? Well, we didn't have an increase in temperature, so it did not take place. Flask C, it did increase, but not as much as A. And this was because A had the added benefit of those microorganisms. It was not sterilized. Those pea seeds were not sterilized. Okay, so there are my variables. The independent variable here would be whether or not the cells respire or not, or those seeds respire or not. Dependent variable, heat given off. So this is what I'm actually physically measuring. 5.2. State two ways in which the reliability of this investigation can be improved. If you have studied your work, reliability deals with 1, the sample size, and 2, the repetition of the investigation. So if I want to improve reliability, I increase my sample size or I repeat my investigation. So it asks us about this investigation and how we can improve this investigation. So I could repeat this investigation. That would be one of the ways. But how would I increase my sample size? I could use more samples. In other words, instead of 40, I could use 80 pea seeds for each of my flasks. That would increase the reliability of my results in that average temperature that would have been produced. Right, so we're going to repeat the investigation there as well for 5.2. That's the second mark. Question 5.3, identify two factors that were kept constant, not what should be kept constant. This means we need to go back to that original context and have a look at what the scientists did in terms of what they kept constant to ensure the validity. Because if variables are kept constant across an investigation, it adds to the validity of an investigation. So two factors that were kept constant in this investigation means they were kept the same. So we need to make sure our answer starts with the same. In this case, it would be the same number of seeds per flask. The starting temperatures were the same, and they were soaked for the same amount of time. Now, we only need to write down two, and there are a few that we could write down here. The only thing that counts against us is the fact that are we showing that these variables need to be the same for all flasks. So we started with same number of seeds per flask, same temperature to start at, and the same amount of time that each seed is soaked for before the experiment starts. Right, so that's question 5.3. Let's look at 5.4a. Provide an explanation for. So this is like why. Provide the why for. Why soaking the pea seeds in water before placing them in the flask? Here we can see that it weakens the tester and allows germination to take place. It softens that tester. So it's going to allow germination to take place. So we soak each of the seeds in water for a certain amount of time so that that is a uniform variable for each one of my flasks. Great. Question B. Why was there no increase in the temperature in flask B? Because boiled seeds will not respire because they have been cooked. They have been killed. Therefore, there will be no heat. Question C. Provide an explanation for the greater rise in temperature of flask A when compared to flask C. So let's revert back. What was the big difference there? Remember I asked you about this before? Right, flask A, there was the possibility of microorganisms still being present on those seeds, and they also respire, which means they can add to that heat that is being produced in the flask. Whereas flask C had the sterilized pea seeds. Why was the flask B included in the investigation? Well, it was included as a control, because how would we know whether or not respiration results in an increase in temperature if we didn't have a flask of pea seeds where we knew no cellular respiration was going to take place? We can compare in each of our flasks then the increase in temperature. So flask B was there to act as a control for comparison with the other flasks in this investigation. Right, Junior Tickies, I hope this has helped in your understanding of how to approach an investigation or an investigative example inside of a formal assessment. I know I've jotted down a few of the answers in shorthand on the page over here, but if you have a good think about what these questions are asking you, you'd be able to structure your sentences quite nicely. And remember, when you're answering a question, get to the point quickly. All right? No examiner or marker wants to read through a long-winded explanation for a question that is worth 2 marks. So get to the point, answer the question as directly and as to the point as possible, basically. All right, that's the way that you are going to make sure you maximize your result at the end of the day. Right, thanks for joining me, Junior Tickies. Do tune in to some of the other videos. Practice those worksheet questions. Have a look at the memorandum and use the theory booklet to prepare for your end of the year exams. Thanks again.